Everyone, and welcome to our video on the structure of cell membranes. This is part of our Module 2 content for the A-level biology, and what we'll be looking at in this video are specification references 2.1.5a, looking at the role of those membranes within cells and at the surface of cells, and 2.1.5b, the fluid mosaic model of membrane structure and the roles of its components. First thing then is we need to understand the actual purpose of a cell membrane. Now, if we think about the most basic principle that you'd have learnt back in year 7, then we talked about this being a barrier. So what we're really looking at there is something that's going to separate different items from other areas. Now, what we can say at A level is that it's separating the cell contents from the exterior environment, i.e. keeping everything that's meant to be inside a cell separate from everything outside the cell. But it also can separate the contents of an individual organelle from the rest of the cytoplasm within a cell. So what we're looking at here is basically the idea of compartmentalization. Now, in our organelles, we can have compartmentalization itself, whereby anything for a particular part of the process is kept within one region, thereby making it more efficient. If we think about how cells function, then one of the key things that they need to allow to happen is for some substances to actually cross from that external environment into the internal one. And that could be obviously the external environment of a cell into the cell, or it could be from the cytoplasm into a particular organelle and vice versa. So we've got to have this ability of substances to cross that membrane, which is what we term permeability. So wherever we're talking about permeability, we are talking about the ability of substances to cross from one side of a membrane to the other. If we think about some of those simple molecules that we've talked about over the course of your biology knowledge, then very small ones, like oxygen for example, will just diffuse through the cell membrane. They're small enough to do that, so simple diffusion works absolutely fine. There are some substances that are going to dissolve in the lipid layer and therefore pass through the membrane. And then there are other substances that need a bit of help, let's say. So they need to pass through a type of protein. And these could be channel proteins or carrier proteins. In terms of the correct terminology to describe a membrane, we should generally term them as partially permeable. And the reason we use this phrase partially permeable is because not all substances can cross it. If it was fully permeable, absolutely everything can cross that membrane. But that's not going to be the case, because what we find is there are certain substances that will not cross from one side of that membrane to the other. So it's partially permeable. One phrase that we are going to have to get used to and kind of upgrade our knowledge and our language a little bit here is how we talk about membranes in our written answers for an A-level biology paper. If you just write membrane or cell membrane, that's a very GCSE type answer or key stage 3 answer. We want you to upgrade this a smidge for A-level. So whenever you're talking about that membrane at the surface of cells, use one of these two phrases, plasma membrane or cell surface membrane. And get into those habits from day one of don't just write cell membrane anymore. We need to be talking plasma membrane or cell surface membrane. Take your pick, whichever one you prefer, go with that, but make sure you are using one of those two phrases in any of your written answers here. In terms of that function then of this cell surface or plasma membrane, then as we already said, separate cell contents from the external environment. It's also, in its partially permeable way, going to regulate the transport of substances both into and out of the cell, because we've got some element of control here. We may find there are enzymes for certain metabolic pathways within the membrane structure itself, and we'll have these antigens embedded within that plasma membrane to actually signal immune cells for various aspects, which we will look at in our Module 4 work on specific immune response, for example. We're also going to find another type of receptor on that plasma membrane, which are the receptors for certain chemical signals. And this is going to be vital in that cell signaling, that cell-to-cell communication that has to happen to bring coordination to all these different cells that then come together in a larger organism. If we then think about the membranes within cells, those membranes around the organelles, that's all to do with separating the organelle contents from the cytoplasm. And this ties into increasing the efficiency of processes. If you know that everything you require for a particular process is contained within an individual organelle, it's all in one region of that cell. We're not having to make something in one place, transport it halfway across the cell to then encounter another enzyme and then transport it all the way back again for the next. Everything is contained within that membrane in a single organelle, a single region of that cell. What we're going to have a look at then is something we call the fluid mosaic model of cell membrane structure. So this is a lovely little diagram showing you what the actual plasma membrane or cell surface membrane looks like in diagram form, of course. So what we can see first of all, we have got our little phospholipids, and do notice how we have two layers of phospholipids. So that means this is going to be termed a bilayer. Bi meaning two, layer, layers. So basically we have a two-layer phospholipid structure. And notice how those fatty acid tails are pointing inwards. The phosphate heads point outwards. That just correlates to what we've already said in the structures of our phospholipids. The heads, those phosphate heads, are hydrophilic. They face towards that aqueous environment. Whereas the fatty acid tails, they are going to be hydrophobic, pointing away from water, so towards each other. What we then have are these different proteins that we mentioned. So we've got a channel protein and a carrier protein, and you can see these actually span the full width of that phospholipid bilayer. So they're providing some kind of an access point across that membrane. We then also have little molecules of cholesterol which are embedded around those fatty acid tails, as we can see. And on the surface we have got these structures, these glycoproteins. So a protein with a carbohydrate chain attached to it that are protruding from the surface of the cell. And these are going to act as those receptors for things like our cell signaling. You will also find that there are some other peripheral proteins also scattered through the membranes themselves. But this gives us a nice little overview of those key components in this fluid mosaic model of our cell surface membrane or plasma membrane. Now, in terms of this phrase, fluid mosaic model, obviously it's a model because we're representing something that is actually far too small for us to see with our own eyes. So we're using something to basically give us a visual representation of it that we can see, draw, understand, discuss, etc. The other parts, the fluid and the mosaic. If we look at the word mosaic and we think about what mosaics are, then if you consider the word mosaic in art, then that's where we've got all these different little pieces coming together to make a larger picture, for example. Now, in terms of applying that to our cell surface membranes, then what we have is a phospholipid bilayer, and then we've got various proteins scattered throughout it. So we've got these various components coming together to make this overall structure. So it's a mosaic. And then the fluid part, because we've got the ability of these lipid molecules to move around each other, and even some of those proteins have certain elements of movement, then it's fluid, because fluids are able to flow and move from one place to another. So we term this the fluid mosaic model of our membranes. Now, when we're talking about the basic structure of a cell surface membrane or a plasma membrane, if ever you get a question asking you to describe or explain the structure, then we're going to go straight in with phospholipid bilayer. That's the first thing you're going to be writing down. It is a phospholipid bilayer. Then you are going to explain what that means. So once we've got the phrase phospholipid bilayer in there, we then talk about hydrophilic phosphate heads facing outwards and the hydrophobic fatty acid tails pointing inwards away from that watery area. So try to remember that when you're writing these, to keep this whole section as a single term in your minds and in your answers. So hydrophobic fatty acid tails and hydrophilic phosphate heads. It's just really good practice to just use that whole phrase when you are talking about these phospholipids and their arrangement, because it means that you're hitting all potential marking points in that simple use of a phrase. So hydrophilic is telling us why it's going to arrange that way. Phosphate heads tells us what it is. Hydrophobic fatty acid tails, again, we've got something to do with the actual functionality, the hydrophobic nature, and we're saying what it is. Don't just say fatty acids or tails. Let's actually use the full phrase to make sure we maximise our marks. Now, because this is a phospholipid bilayer, then what we find is this is going to be a barrier to most water-soluble substances. And this is going to be important for our living organisms because we don't really want to lose large quantities of water-soluble substances from inside that cell. Remember, it's quite an aqueous environment inside cells. And therefore, we don't really just want it leaking all of these useful substances to the surroundings. We want to keep them inside the cell for the cell to actually utilise. Therefore, by making the actual surrounding of our cell, that cell surface membrane or plasma membrane, out of something that acts as a barrier to these water-soluble substances, it does that. It reduces that loss. It is worth bearing in mind, though, that we do have to transport things in and out. Therefore, those transport proteins come into play. So we talked about two types in that little diagram earlier. The channel proteins span the whole bilayer, and these are all to do with allowing ions to pass through. We then have carrier proteins, and these ones are actually going to be changing their shape to actually enable the movement of certain chemicals. So they don't have a fixed shape. What we find is that as they're then transporting a substance across it, the shape of that carrier protein is going to be altered. So it undergoes this conformational change to move substances from one side to the other. We mentioned our little cholesterol, and the role of our cholesterol in our membrane is to increase fluidity. Now, if temperatures drop low, then what happens is those phospholipid molecules are going to start to move closer together. They're going to become more condensed as a structure. If they become too closely packed, then the membrane itself is going to be quite rigid, and that means its functionality can be lost. So we need to stop that. What we do is we then incorporate those molecules of cholesterol amidst the fatty acid tails there, and that means that as the temperature drops, it almost acts kind of like a spacer. It prevents those phospholipids from becoming too tightly packed and therefore maintains fluidity of our membrane. We also find that cholesterol has a role at the higher temperatures too, in the fact that it actually stabilizes the membrane and stops it becoming too fluid. So when we're thinking about cholesterol itself, it's this really important part of a plasma membrane to do with maintaining that mechanical strength and the stability of the membrane itself. So cholesterol is one of those key components that we must remember. On the outside of the plasma membrane, we have this structure called the glycocalyx. And the glycocalyx is basically a carbohydrate chain attached onto something else. So we've either got it attached onto lipids to form glycolipids, or attached onto proteins to form glycoproteins. So glyco just tells us carbohydrate, and the second part of the word there is telling us what that carbohydrate is attached to. So glycolipids, carbohydrate stuck on a lipid, glycoproteins, carbohydrate stuck on a protein. And these are going to be protruding on the outside of the plasma membrane. So they're basically sticking up from the outside of the plasma membrane itself, And that means that we've now got this structure which can act as a receptor molecule in that cell signaling process. So what will they be binding to? These could be hormones and neurotransmitters. We could see that they are receptors for that process of endocytosis, whereby we're going to be moving substances into our cell. They could be involved in cell adhesion and stabilization. So obviously cell adhesion is where they're going to be sticking to things. And it could also be to act as an antigen for this cell-to-cell recognition. So things like our immune system, where we need to be able to recognize our own body cells as self, then these are going to actually act as an antigen so that our immune system cells know that's self, that's part of us, we don't attack that. It is worth bearing in mind that when we come down the route of these specialized cells, then they've differentiated to have features for a certain function. So what we find is a generic cell has certain features, but when we're undergoing that differentiation process, then clearly they've got a very specific role to carry out inside the organism. So that means they're going to need certain adaptations to make them effective at doing that. So something like our white blood cells, they will have plasma membranes with protein receptors, all to do with recognizing antigens on those foreign cells. So they can identify which are foreign cells and therefore which cells need to be removed from the body in our immune response. If we think about plants, we could use the example of the root hair cells. And these ones are obviously designed by the plant for taking up substances from the soil. So through those root hair cells, we are going to need to actually take up all those nitrate ions from the soil through that process of active transport. So we need lots of carrier proteins to allow that active transport to take place. I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when we next upload another video, and head on over to the website where you can find a range of other resources hopefully designed to help you improve your A-level biology knowledge and scores.